in the world. So let's continue, let's move on. And I am pleased to introduce Rafaeli from the Hebrew University Israel that is going to talk to us about features of hippocampal astrocytic domains and their spatial relation to excitatory and inhibitory neurons. Ron, please go ahead.
Thank you. Okay, so first of all, I want to thank you so much for inviting me here to have this lecture. As you mentioned, my name is Rafaeli, and I'm from the Hebrew University of Jerusalem. And I'm going to talk with you about, let's see if it's worked, that's right. Yes, so I'm going to talk about features of hippocampal associative domains and their spatial relations to excitatory and to inhibitory neurons. This is from a paper that recently published paper from our lab, which actually presenting the astrocytic interaction or spatial interaction between excitatory neurons in astrocytes and inhibitory neurons in astrocytes. So as I'm sure you all know, and as was shown before in previous lectures, astrocytes were noticed and mentioned already by Ramon Cajal, a cell which enveloped these avocado-shaped neuronal cell bodies, therefore probably have some important role to play using this spatial interaction. And nowadays, it was already beautifully established that astrocytes do not only assist to the neuronal metabolism or find satisfaction with being a part of the blood-brain barrier. Using the tripartite synapses and other morphological properties, astrocytes' arsenal is now known to be well-equipped. Astrocytes can change the ionic concentration, can influence glutamate and GABA, etc. But they can do so much more. So recently in the lab I'm working in, the Goshen lab, two breathtaking papers were published showing the important role of astrocytes in memory formation and consolidation. And the first paper, Adamski 2018, she presented that when hyperactivating C1 astrocytes during acquisition of a memory, recent recall improves while remote recall remain untouched. And the second paper, Fkolot 2020, actually further presented that inhibition of C1 astrocytes harm the remote recall, but do not interfere the recent recall. And these two results enable us to understand that memory timelines is not a straight line as was thought, but rather two different pathways bound to one another and defined at the moment of acquisition. This shocking result, it was kind of a shock to see that this is not a linear track of acquisition and then recent and then remote, etc., but actually two parallel pathways. So it was provided by astrocytes investigation only. Okay, so only investigating and manipulating astrocytes, this kind of a new shocking neuronal results was observed. Since it appears that they influence those two parallel pathways differently, the astrocytes. And so they can create de novo LTP, and their influence on the neuronal synapses can affect cognitive processes such as memories. But how do they influence? I mean, how does the astro-neural interaction happens? So we speculated that it has something to do with their domains. So let's first talk about what is associative domain or territory. So in the hippocampus, as well as in other many brain regions, astrocytes come in distinct domains, territories, and the processes do not intermingle, in opposed to the neuronal processes. And so it makes sense to assume that, that nearby neurons, at least their somata, are bound to the nearest domain more than further surrounding domains. The same assumption can be also accurate when looking at the synapses level, though we're not going to mention it right now, but we can talk about synapses domain and somata domain. Though many studies have investigated the tripartite synapses, too little or not enough is known about the neuronal somata and its interaction with nearby astrocytes. One study from 2017, and only one study as far as I know, looked at the number of cells bodies in each domain, in the striatum and in the stratum radiatum of the hippocampus. And at least for the hippocampus, the SR, the stratum radiatum lamina, is by far not as crowded as the pyramidal layer itself. All the more so if you want to say something about the functionality of domains, when the main output source of the hippocampus is actually the pyramidal neurons, the pyramidal layer neurons. And so this lack of information regarding how many neurons are enveloped by each domain, it occurs mainly because it forces the researchers to tag all neurons and all astrocytes. I mean, sparse labeling will not give us relevant number of neurons per domain. Not if it's sparse labeling of neurons and not if it's sparse labeling of astrocytes. On the other hand, tagging all neurons, since some brain regions and the hippocampus and the striatum is part of them, it will eliminate the option of targeting single synapses, just because it's just too crowded. So even though the study of associative domains have expanded and become more sophisticated, not enough or almost not at all is known about the importance of territories, at least when you're considering the neuronal somata, and at least when you're considering the hippocampus. So what we did, together with Adido Ron, for another student from my lab, we decided to investigate the astroneurospatial interaction. And again, I'm highlighting the spatial interaction in the C1 and in the amygdala, while targeting the whole morphology of the astrocytes, but highlighting the neuronal somata. And again, not exons and not synapses and everything, just the somata of the neuronal subtypes that we will talk shortly enough, and all the astrocytes. Using transgenic mice and viral infections, we fluorescently tag 4 types of neuronal cell types and astrocytes in 4 different groups of mice, of course. Firstly, we tag all excitatory neurons and astrocytes, and secondly, we tag the inhibitory neurons and nearby astrocytes. Post-mortem, we use declarative procedure, as I hope that you're familiar with, but you can create a full, thick, transparent samples. And so we were able to image thick slices of hippocampus and amygdala. Eventually, what we got is an imaged 3D information of all astrocytes. You can see it's more than 97% of all astrocytes were tagged, and all excitatory neurons with more than 96% of excitatory neurons, and pretty much all the inhibitory neurons, the somatostatin, the VIP, or the parvalbumin. And to go over the pipeline we did, so we inject mice with viral vectors in order to tag all relevant cells, again, hippocampus, C1, or in the amygdala. We made thick hippocampal samples transparent using clarity procedure and imaged them using confocal and 2-photon microscopes. The image was later reconstructed in the Imaris software, where for astrocytes, all morphological information reconstructed, and for the neurons, the centroid coordination was extracted. Those matrices were then exported into MATLAB for further analysis. So this is just basically the pipeline that we did. In this movie, you can see a slice-by-slice information from a 1 millimeter thick image sample. Pay attention to the amount of detail presented. All processes of all cells are fluorescently tagged. Here you can see now what happens when all the information is forged together. Again, you can see that all astrocytes are fully tagged and almost none of them is truncated. And it's super important because usually when you're using 2D information, some of the process will get out of the images because it's too much of a 3-dimensional bushy kind of thing. And so having this information allow us to commonly ask questions regarding the entire associative population. If not all astrocytes were tagged, the questions that could have been answered were only limited to specific interaction, a specific synapse or a specific cell. And again, we asked the question in this paper, and we ask about all astrocytes and all neurons in specific cell types. So first of all, we need to know that after we imaged all astrocytes, we can personally care each and every astrocyte and to be sure that all astrocytes are correctly reconstructed. So the first thing we did was to characterize the associative morphology properties. We infected, we cleared, we imaged and reconstructed fixed slices of hippocampal astrocytes. And while each astrocytes were carefully reconstructed, you can see it in E and F and G, you have the fluorescent, the information, the channel or the flow of information in magenta, and then in F you can see the whole astrocytes fully constructed, and then in a compact representation to work with Imaris, with MATLAB, sorry. And in here you can see how strict are the domains. In the left side, you can see how strict they are, and you can clearly separate this one from the other. And you can see how beautifully the imaris, the filament feature, are beautifully separated too. Another movie, and this movie presents a 3D view of another thick hippocampal C1 astrocytes, where clearly most astrocytes are, again, not truncated. And then we reconstructed each and every one of them, creating a fully reconstructed C1 cubes. So for further on, when I'm talking about a cube, a fully reconstructed cube, it's a couple of hundreds of astrocytes within it. The first one, so first we measure the distance of each associatic soma to the surface of the pyramidal layer to define the lamina belongings. Okay, we have 3 different laminas, as I'm sure you know, or I'm hoping that you know. So we have the stratum radiatum and the upper part, the lower part in here, and then we have the pyramidal layer and the stratum oriens. So first of all, we saw the distance from the pyramidal layer and define the lamina it belongs to. And then we analyze distribution of associative domain of the associative soma from 55 thick cubes. So we got 19,245 astrocytes to analyze. And we saw that, first of all, the density. So the density in both stratum radiatum and the stratum oriens increases toward the pyramidal layer. And inside this layer, density drops toward the middle from both sides. The average size of hippocampal astrocytes starts to drop at around approximately 20 micrometers from the pyramidal layer in both stratum radiatum and the stratum oriens, and continues to decrease toward the middle of the pyramidal layer. Associative orientation. Okay, so we use the longest vector. So we found the longest vector of a specific astrocyte, and this was the angle of it. So it was calculated relative to the surface of the C1 pyramidal layer midline. And it seemed that at the lower part of the C1, as well as in the middle of the pyramidal layer, the astrocytes are more perpendicular. Okay, so we can see the shift from around 25, 30 degrees to 40, 50 degrees. Sorry. Okay, so another thing that we did, so in order to investigate the spatial relation of astrocytes and their surrounding inhibitory neuronal content, we co-labeled inhibitory neurons and astrocytes. We we imaged for PV and VIP and SST, somatostatin. And from the very first look, just by looking at the figure, it seems that SST neurons comes in very close proximity to the neighboring astrocyte soma. Again, I'm talking about soma to soma. This kind of phenomena, we called it hugs. We checked if this phenomena is due to the differences and the different densities of the neurons and astrocytes in the hippocampal lamina, and I will explain. If you look at D and E, sorry, so you could, if you look at D and E, you can assume that only by chance the VIP neurons should be farther away from the nearest astrocytes, since they are abounded in the pyramidal layer itself, where you have a lot less astrocytes anyway. The SST, the somatostatin, on the other hand, are mostly found in the oriens layer, where the astrocytes are larger and more dense. Therefore, or to answer this kind of limitation or kind of thing, so we shuffle the information using permutation to see if the hugs observed are purely mathematical or is it more to it. And we found that this phenomena is not strict to the distributions of cell, but rather special and probably play a role in the functional properties. And again, I will highlight, we did not check for functional kind of things, but we did find something which is more than you would expect. So probably we are guessing or educatedly guessing that it has something to do with the functionality. And we highlighted the proximity between somatostatin and the nearby astrocyte soma, but you can also see in FNG that the VIP are also kind of tend to come farther away than you expected. So the SST is closer than you expected, and the VIP is farther than you expected to the nearest astrocyte soma. And so since the vast majority of C1 neurons are excitatory, or the more so when looking on the output information from the C1, we wanted to characterize the excitatory neuronal content in those associative domains. For that, we co-labeled astrocytes and excitatory neuronal nuclei, where all the neuronal nuclei are represented as spots containing, again, XYZ coordination, and all the astrocytes were fully reconstructed. And the average amount of excitatory somata cells in each domain is approximately 14. And the size of the domain is not the dominant feature to predict the number of cells within it, but rather its place relative to the pyramidal layer borders. I mean, astrocytes deep in the pyramidal layer envelop the largest number of cells. It's a bit of a surprise since we saw before a few slides ago that the astrocytes adjacent to the pyramidal layer are much larger than the astrocytes within the pyramidal layer. And we also measure the same thing, the spatial interaction of astrocytes domain and excitatory neuronal and non-excitatory neurons in the amygdala using the exact same methods. And we found that 4 to 5 neurons in each domain were found, which come in one hand with our knowledge that cells in the amygdala are sparser than the hippocampal pyramidal layer neurons. And nonetheless, this difference may influence or hint some the way that astrocytes may affect each region. Perhaps to do it differently. So conclusion, to sum up, almost 20,000 astrocytes were analyzed regarding their location, their densities, angles, sizes, shapes, etc. And actually we have a huge bank of information. We can decide to do whatever we want. We have 20,000 fully reconstructed astrocytes to play with. The second thing that we did is associative domains and their inhibitory content were defined, finding the somatostatin hugs, also finding the VIP to be relatively farther away than expected. And then the third thing that we did is that we found is the associative domain and their excitatory content. And so they were defined and in the hippocampus and in the amygdala. And this so-called basic information, actually, this basic information of how many neurons in each domain, especially for the excitatory ones, it actually fills a void that until now was missing. I mean, asking how many neurons are to be found in each domain, it's something that you have to know before searching for changes during functional processes or biological or even computational manipulation. If you do some manipulation and find it to be 16, you need to know it's different than the 14 that you're assuming it to be. And it's written in the last sentence, it cannot be achieved by the ratio of astrocytes and neurons. If you have 10 astrocytes and 100 neurons, you cannot assume that you have 10 neurons in each domain. We already saw that even by the lamina, it's differently distributed. Lastly, so I just want to see in the chat.
Don't worry, I'll take care of the questions.
Sorry. So I just want to thank my group, first of all to Professor Bar Goshen, my mentor, my PI, to Dr. Tirza Kreisel, it's the lab manager, but actually like the wizard that can do everything that you want. Adi Doron, which we did this work together, and all the other lab members. And now it's questions time.
Thank you very much for this very interesting talk, Ron, and beautiful data. So for those that arrived or that might not have heard some previous talks from the workshop, just a reminder that you can write your question directly here in the chat or just write question and we will unmute you for questions. And I see here already one question from Kerstin. So Kerstin, please go ahead.
This was really wonderful talk. I really love the data. I was wondering, due to the preparation method, like the clarity, do you expect any volume shrinkage due to that? Because of course this might affect your data at the end.
So actually, exactly for that reason, I mean, it wasn't presented, but we did this kind of validation. We actually, so it kind of go into details. So because some refractive index matching solution, the less solution that you put the brain in, the completely transparent brain in, some of this solution can swell the sample and some can shrink it. We worked with Rapiclear at the time, and it kind of swell the samples. So we kind of do it like we did for a few hundreds hippocampal. We compare the non-clarity and the clarity slices to see how much it swelled. And it swelled the same thing from the X, Y, and Z. And it swelled about approximately 1.8 as much. So if you found it to be 20 micrometers, so we knew it should be relatively 11 micrometers. You know what I'm saying?
Mm-hmm. Yes.
Okay. So we highlighted it in the paper as well and tried to address it also in the data. And by now, also if anyone else is working with clarity, if using clarity specific Rapiclear and not just Rapiclear or Focus Clear, whatever, just different kind of names for the less solution to image in, then it actually shrink it back to pretty much the same size. So the brain, the sample just go back to exactly the same size, or pretty much exactly, and then you can just neglect these changes in the volume.
Okay, thank you.
So no more questions in the chat for now, so I will go ahead and ask my questions. So first of all, technical question. I was wondering what was your resolution in X, Y, and Z for your reconstructions?
Resolution. So it
yeah, spatial resolution.
So in we used the confocal and the 2-photon. The confocal was not as good just to limitation of the objective that we used, and it's 600 over 600. I don't have it on my laptop on my desk right now.
Order of magnitude is good.
But the 2-photon was extremely good. I mean, I can tell you that the numeric aperture, for example, was 0.94. And it was, I'm not sure exactly how much, but it was very good.
Okay, okay. I'm looking forward to hearing more about those reconstructions. I was wondering, so you were showing those neurons that were in each astrocyte domain, and I was wondering if you quantified which compartment of the astrocyte was interacting with those neurons and with which compartments of the neurons. So if you characterize the spatial relationship between neurons and astrocytes at the compartment level or not yet, or?
Not sure. I mean, we did some speculation. I mean, I'm taking a bit aside of what you asked. I mean, if it's going too far, but we want to make sure, because you can look at, I mean, you can decide that if you want, first of all, we assume that neurons should be combined or obligated to only one domain, because you do find some on the border between two different astrocytes. And we try to do some different, to look what happens if you only look from the distance from the soma of the astrocytes to the soma of the neuron, or what happens if you look at the edge, I mean, the nearest dot. You have the soma, and also in order to fully reconstruct the astrocytes, you have a lot of, not the main spot of the soma, but a lot of dots, like a cloud of, which from that imaris afterwards build the whole filament. So you need to look which is the closest. I mean, you're taking like the 20 or 50 closest dots and find most of them are bound to this territory or the other. And we find no really, and so we're gonna do a lot of speculation, what will be the best solution or option to look at it. And we find actually not that differences in the end results.
Okay.
As
very interesting.
Okay, sure.
And I'm looking at the time, maybe. So yeah, one more question then. So you said that for the moment you didn't look at the function, but maybe you could speculate and tell us a bit more about your ideas and how astrocyte morphology could impact the function and interactions with neurons.
So actually, even though the paper was actually, I mean, the main point of the paper was, I think for the beginning, the most important thing was just to define how many excitatory neurons are in each domain. Actually, I think one of the most beautiful results that we got are the proximity to the somatostatin neurons, inhibitory neurons. And afterwards, it was kind of a shock. We just looked at it, expected to see nothing important, just to have it done. And we saw this very close proximity. And it was very robust in a lot of, like in, I don't know, 10 different tubes. It was very robust that they are come in close proximity. And then we actually looked at the literature, and we saw that also in the cortex, we have some paper that talked about not the somatostatin, but subgroup, undefined subgroup, a group of inhibitory neurons that are coming close proximity, somatosoma, etc. So we can speculate that there are somatostatin. We need to further check it. And I'm guessing that, as at least for the hippocampus, the somatostatin are at the stratum oriens, which they are kind of get the information that comes from the outside, from the cortex to the hippocampus. They get the information and they can control the information that's coming to the pyramidal layer as they reach the pyramidal layer, while the VIP and the PV are controlling the internal circuitry inside. They are in the pyramidal layer and they can play with it. So this close proximity of the astrocytes and the somatostatin can maybe play off the how do I want to get this information, not how I want to handle it with myself, but this is something to do maybe to the PV and VIP. And again, it's purely speculation, I don't know. But the somatostatin actually can control the information. Maybe you can tell, like, the astrocytes can tell him, right now, I don't want to get a lot of information. I'm in the middle of processing something else, or something similar to that idea.
Thank you. That's very exciting. So we have one more question from Annalisa. So I'm gonna unmute you.
Okay. Yep. Got it. Data. I was thinking about this proximity between astrocytes and somatostatin into neurons, and I was wondering whether the big question is why, right? What do they do? And have you thought about them perhaps preventing glutamate spill in? I always think about astrocytes as cells that have, glutamate transporters densely packed in many of their domains. So rather than preventing something coming out from synapses contacted or formed by somatostatin into neurons, will they actually shield those synaptic connections from any glutamate spilling in from adjacent excitatory synapses?
So again, you need to shut, okay, yeah, okay. These kind of speculations will be extremely purely speculation, because in my lab, in the lab I'm working with, we're not looking at this kind of resolution or this kind of, I never looked at synapses. I mean, I went the other way. I looked at, I did clarity. I can see the whole brain. I can see the whole astrocytes, etc. But I chose not to see synapses. So just to, I'm thinking how to put it into the world, but the kind of research, the research that I'm doing is kind of, unfortunately, neglecting this kind of resolution, because I want to see the whole astrocytes. I want to see the clear brain. I know that I will not be able to see synapses. So I'm just as smart as any other guy here, just to speculate what will happen, which obviously can do something to that. As I showed you from the paper from my lab, actually in 2018, of course, you can create de novo LTP, and you can inhibit information to come across. And astrocytes do it. I don't know how, but they do it. So it's kind of makes sense. I can play with, I don't know, potassium, calcium, etc., of course. But yeah, sorry, I have zero knowledge of speculation regarding this kind of resolution.
Okay, well,
Okay.
Thank you very much, Ron.
Thank you.
Thank you again for this very nice talk. Can you please stop sharing your screen?
Yes.
Thank you. So this is now my great pleasure to invite our last speaker of this workshop, who is also a co-organizer, as you all know. So Kerstin Lenk from the Graz University of Technology in Austria, and she's going to talk to us about computational models of interactions between neuronal and astrocytic networks. Kerstin, please go ahead.
You should see my screen. Yes, hopefully.
Yes, we do, and we can hear you.
Okay, thank you very much. So thank you, Audrey, for the introduction. And as Audrey said, I'm at TU Graz now in Austria, and the work was mainly carried out actually in Tampere in Finland. So as we heard now a couple of times the word tripartite synapse, presynaptic neuron and the postsynaptic neuron are enwrapped by an astrocyte, and these form like really large networks together. And the same image as Ron actually. When astrocytes connect to neighboring cells, they form this non-overlapping domains and form gap junctions where they exchange IP3 and other ions. So this is one prerequisite we have for our model. And as has been challenged as actually yesterday by Laszlo, we have in the model different dynamics for neurons and astrocytes. So at the moment, we assume that astrocytes are much slower than neurons, so rather on the 100 milliseconds range. And but it would be actually interesting, I have to say, to change that and see how the model would behave on this. And what we have also added, but which is also hugely under debate, is if clear transmission is happening. So there has been in 2018 this dual perspectives papers, or like two papers, of course, and one against and one for clear transmission. And there has been also by the lab of Alphonse Rac one paper where they actually show that one astrocyte could actually release two kinds of clear transmitters here, glutamate and ADP. We use that also for our model, also in the knowledge that this might be not actually the case. Just to show you what data we are using. So at the moment, it's mostly multi-electrode array data, so we can capture the electrical activity of neurons here. We also partially use calcium imaging data from neurons and astrocytes, and of course, like all modelers, we use the existing parameters from the literature. So then we form our equations, and then of course, like as seen also by others, we get hopefully similar results as in the experimental data. And what we also hope to do at some time point is predictions. Then this is the model we are using at the tripartite synapse level. So at the moment we have only that the excitatory neurons are connected to astrocytes because there hasn't been, like when we built the model, there hasn't been too much information on how GABAergic neurons are interacting with the astrocyte. And this is based, which was actually very interesting for me yesterday, also on a probabilistic model as Yukiko has shown. And so we have an incoming spike. We have a glutamate released towards the synaptic cleft, which is either taken up by the postsynapse or by the mCLR receptor at the astrocyte. This triggers then the IP3 release from the endoplasmatic reticulum, followed by calcium release, which might then end up in the calcium-induced calcium release. And then we have towards the presynapse glutamate release or ATP release, and counterbalancing the whole process adenosine towards the postsynapse. And we also have included gap junctions, so two neighboring cells are connected to each other, and they are indirectly exchanging calcium. So like IP3 is exchanged through the gap junction and triggers then calcium release in the neighboring astrocyte. I just want to mention this. So this is built on Maurizio De Pita's model, and we also have included from the same lab of Hippocampal Jules Lalouette's model of how the calcium dynamics are working in the astrocyte network. And interestingly, it's so that in astrocytes, the more connections we have, there's actually less calcium spread, which is different to what we have in neurons. Then then we built our model with first 100 neurons without any astrocytes there, and we see a regular spiking in the neurons. Then we added 80 astrocytes. As said, they are just connected to the excitatory neurons, and we see that we have this burst-like behavior, just as we were expecting. So like one of our hypotheses was that actually astrocytes are responsible for bursting. Then we wanted to know how the number of astrocytes in the network is influencing the neuronal activity, because of course, as you might know, there's different amounts of astrocytes in different brain areas and across different species. So what we did here is we used 200 excitatory neurons and 50 inhibitory neurons, and they had a connectivity of around 30%. And then we either had no astrocytes, 10%, 20%, or 30%. We simulated a spike train of 5 minutes. And here it's shown like that the neurons could form long range connections, and the astrocytes rather short term, not short term, but short distance connections. And here in green, you see a virtual multi-electrode array, as I've shown before. In green, we have the neurons distributed. They are the same for all the 3 networks. And then we have 10%, 20%, and 30% of astrocytes. And you see in red also their connections. I just want to highlight, I learned yesterday that this paper where we have published this is the Frontiers in Computational Neuroscience editor's pick for 2020. I'm actually saying that because I think it highlights how our field is becoming important. Then what is happening at the tripartite synapse? So this is one example from one tripartite synapse, and we see here the presynaptic activity and the amount of glutamate released. And we see that the astrocytic IP3 is following that behavior, as well as the local calcium with a small delay. The diamonds, they indicate the points where glutamate is released. So we have then here the glutamate concentrations released to the presynapse, and we have at the bottom the indication how the postsynapse reacts to that. This is the resulting neuronal activity. So we have the spike rate, so the action potentials per minute, the burst rate, so burst is a cascade of spikes also per minute, the average spikes per burst, and the average burst duration. And we first modeled how the network behaves when there are no astrocytes are present. Then we connect in the model the astrocytes with the excitatory neurons, but the astrocytes are not connected with each other, so they form no network. And then we have the full network with 10%, 20%, and 30% of astrocytes. We also applied different noise levels to the neurons to see how this influences the spike activity. As you can see, like we have in a healthy range, the spike rates, so not pathological spike rates, it's increasing a lot when we connect the excitatory neurons to the astrocytes, but the astrocytes don't form a network. And actually, the activity goes down when the astrocytes also form their network. We have a similar behavior for the burst rate. So you can also see in the insert that the burst rate is going slightly up for when the amount of astrocytes is increased. The average spikes per burst remains quite similar throughout all the simulations, and that means that the burst duration actually goes up when we have more astrocytes. So this is usually not the way we are going, but we did actually the experiments afterwards, which was really interesting and exciting. And this is work by Barbara Canoggi and Annika Ahtiainen from Tampere University. So they're working at the moment on this data. And what they have done is they really defined the amounts of astrocytes in the neuronal culture. So we always used the same amounts of neurons. And we stopped the proliferation of the astrocytes so that we could have on the multi-electrode arrays 10% astrocytes, 20, 30, and 50%. And then we measured the neuronal activity. And as you can see here already, and I have one more slide on this, the burst duration is actually also here slightly increased when we have more astrocytes. So this is the quantification. So you have here indicated by the different colors. So NS means it's a theoretically pure culture. Of course, we still have some astrocytes in there. And then we added 10, 20, 30, and 50% of astrocytes. Here are the days in vitro. The networks have been cultured on the multi-electrode arrays, and you see an increase of activity when for the NS cultures, so for the neurons only cultures, and then we have actually this down regulation also when we have more astrocytes present. There's a slight increase throughout the cultures of the spike rate and a decrease when we have 50% astrocytes. We see that a similar behavior for the burst rate, even if it's not like as pronounced as for the spike rate. But still we have like higher activity in the NS cultures. Then what Barbara did is also, she wanted to see if we kind of induced epilepsy on the multi-electrode array. So she simulated that. So she used this virtual multi-electrode array again and stimulated one area in that. And the result is that we see this huge amount of firing during the stimulation period. And what we wanted to see then, of course, is also how the astrocytes react on that. And no surprise, there was a huge calcium release. Then she also did further simulation. It was basically confirming what I just said before. So here also you see that the stimulus is driving the neuronal activity, and with a small delay also the astrocytic calcium activity. Then one recent paper which went more towards graph theory and molecular communications. We tried, or we actually simulated a 3D network. So we went from the planar one to 3D. Here in red are the neurons, as you can see, and in blue the astrocytes and their connections. And what we did is we played with the distance between astrocytes in this network. So, but we changed it uniformly. So, so either we changed it to 70 micrometer, or to all astrocytes, or to 100 or to 120. And K is like from the graph theory, the mean degree, indicating the number of gap junctions. So it's increasing with the distance. Then we have the shortest path, L, which is the minimal number of gap junctions one has to cross to connect two astrocytes with each other. This is subsequently going down. And then we also measured the active astrocytes. And it was interesting that we had the highest number of active astrocytes when the ranges were short, and then it decreased for the larger ranges. We also see that the spike rate is increasing until about 100 micrometer distance between astrocytes, and then it's slightly decreasing or stabilizing at this level. Same for burst rate, and the burst duration is also following that that way. This is showing the 3D network again, and the spike rate, so the average spike rate. And you see that it's first of all increasing mainly until 220 micrometers. And we see this kind of hub cells in the middle, so which actually, so the neurons induce a higher, show a higher activity in the middle of the cell culture. Then we have one work together with the same model, proving several hypotheses of schizophrenia. So this is, we have submitted this, it's under review, so I'm really excited about it. I'm happy to talk offline about it. So we tried several, schizophrenia is very difficult disease, where there are a lot of hypotheses how it's working. You also see different, like different papers indicate different neuronal, especially reactions, and so we try to study that and make some sense which hypothesis could be valid. And then to wrap up, so we have this neuron astrocyte network model. It was one of the first ones at the time. And we connected several hundreds of excitatory neurons to one astrocyte. We saw that the astrocytes have a profound control over the neurons. And we saw a prolonged burst duration of neurons, but it restricted the hyperactivity. What you probably have noticed, we have not included all the pathways that are known by now. And that's, of course, one thing we would like to add, but we actually are going at the moment one step back. Because, of course, there's already a lot of equations and parameters included in that model, but we realize we have to include much more to, for example, study diseases like epilepsy in more detail. So we are working at the moment on this model, which includes also morphology and other ions than calcium. Then I just want to point out for the ones who would like to get a little bit more familiar with different models of astrocytes and also in the network level. We had this review from 2018, and there are also two wonderful reviews from Tina Manninen, who's also in the audience, which might also help you with that if you want to get more familiar with that. And with that, I close, and I'm happy to take questions.
Thank you very much, Kerstin, for this amazing talk. A very nice way to end this this workshop that I hope everyone enjoyed as much as I did. So I'll take any questions. Please write in the chat your question if you have one or ask to be unmuted. We have already one question from Alan that is asking whether the calcium dynamics in your model is local near a given synapse or neuron, or is it global within the whole astrocyte?
It's rather global in a way because we don't have any like, which you might have maybe got the impression that there's any morphology in the astrocytes or in the neurons, but they are rather considered as point models. So there's so we have the incoming glutamate, which triggers then the calcium, and it's uniformly distributed throughout all processes or like connections it has to others, to other neurons and astrocytes, of course.
I have a follow-up question about this, except if Anna has a follow-up question. Okay, so I'll follow up. Yeah.
Yes.
I'm going ahead. So I was wondering then if you could speculate what would be the effect of adding this morphology, so the diffusion of the signals within the cell, if you have any ideas how it would impact the activity at the network level. It's quite a tricky question.
It's really a tough one, actually. Oh my God. I mean, you mean just using the IPA3 dependent pathway, right? Or would you also consider then other pathways?
Yes, I'm just wondering because for the moment, if I understood correctly in your model, as soon as you have a calcium signal resulting from neuronal stimulation, it's everywhere within, it's as if it was everywhere within the cell, which means
exactly.
That
so
it has higher probability to diffuse within the syncytium, right?
I think that might change then, of course, like that you actually have, it might be that you have actually less calcium propagation at the end. I think that might actually happen. Yeah.
Or it could be as my model at the nanoscale, and actually at the end you have the opposite effect, and because you have more compartmentalization at the end, it increases the size of the signals so that you have more propagation.
I'm happy to try out. So
yes, looking forward to the morphology.
Might just need 10 years.
Yes, most probably. So thank you again, and maybe let's go on to the discussion, as we have no further questions for Kerstin.
Thanks, Ala.
So shall we have a combined discussion between Kerstin and Ron, or Kerstin, do you want to go ahead and have more broader discussion about the field and the future of the field?
If there, please feel free to ask questions to both of us. Let's wait a couple of seconds, and otherwise we. Okay.
Yeah.
So
let's move on.
Yeah, thanks, Audrey. What we would like to do is, so we would like to ask all speakers if they, also from yesterday, if they could put up their camera if you want to. So we would like to have a little bit more extended discussion, and would actually like to ask for, we have 2 directions of questions, and we would be happy to discuss them together. So if you're able to, and if you want to, we would be happy if you could get them online, and of course also our organizers. Perfect. Thank you. So, but maybe if others want to join in, that would be also created some. Let's get you all in here. Yes. So one thing, what Renaud mentioned earlier, and I was really wondering about this as well. So he mentioned this Frankenstein models. And so that we basically have the parameters from all kinds of animals. So I did once the fun part from Maurizio De Pita's model to check actually where the parameters are coming from. And I found a whole zoo, but for example, no, if I remember correctly, no human parameters. So I was wondering, do you think it's okay? Do you do you think, what do you think, how could we overcome this? Also, for experimentalists, if you have any thoughts on that, how we could work in this regard together. So I would be happy to hear your opinion.
I just want to say that I'm not sure if you're familiar with this paper that they inserted human astrocytes into mice.
Yes.
And so I think hopefully I'm not going to misguide you, but they actually perform better, right? Cognitively?
Yeah, it's Han 2013 or 12 or 13. Yes.
So first of all, I guess it can go in two different ways. Mice are resilient enough to have something from a completely new animal, and survive with it and actually handle with it, and also kind of look like you can at least find some similarities, and you can assume, like, whenever you're using animals, you want to assume at least that it have something to do with humans. But I mean, on the other hand, you do know that they are completely different. I mean, just by looking at the morphology, they are completely different. And if you look in the morphology and you see a lot more processes and farther away from the soma, you can assume that you will have a lot more gap junctions and interaction and excitatory and inhibitory synapses within it that you can interact with. So you're gonna at least remodulate it all over again when you're trying to look at humans. But it's a good place to start, at least until we won't have any transgenic humans. This is what you can do.
Yeah, I think there's even if you had human data or a species specific degeneracy.
You're frozen, Suhita, I think.
Okay, I thought it was me.
No, I think Suhita is frozen.
Yes, of course, I agree that you have to start somewhere and then find other ways to constrain your models. Can you hear me?
Yeah, now you're back.
You were frozen, so we didn't really hear what you wanted to say.
Okay, so I was talking about a degeneracy, intrinsic degeneracy in parameters in any system, right? And especially in neuroscience. So there's a whole wide range of parameters within a species across brain areas, and so there is no one magical number. I think what modeling allows you to do is play around with this, and so it's a great place to start. And to have more accurate predictions, meaningful predictions, you have to find other ways to constrain your models. A higher level constraint, a higher level experimental data that you can use to bootstrap to say, this is one of the ways in which the system has found its solution. And so instead of, I think, thinking of it as a bug, I would sort of think that it allows you to explore more. So probably gives you more insights. Because you are forced to sort of, you're forced to constrain your models in different ways. And when you have to do that, I think it's arriving at a new insight.
Go ahead, Renaud, if you would like to.
Yeah, I think it's really important that we make sure that whatever models we develop are match biology as much as possible, especially at this stage, because I think, unlike in traditional computational neuroscience, where there's a good quantitative or qualitative understanding of what neurons do, I think it's far less clear what, for instance, astrocytes or other glial cells do. And so we need to be sure that whatever model we develop, they match the biology as closely as possible, because otherwise we run the risk of developing models that are very nice in papers, but that if that are completely describe fictional brains, right? And so I think it's really important, until we have a better conceptual understanding of what astrocytes do, for instance, and how they play, what role they play in neuroglial networks. I think it's really important that we stick to experimental data as much as possible, even though it's really usually quite difficult. Because I think computational neuroscience it's less of an issue when you do just neural networks, because maybe the behavior of neurons is more stereotypical than the behavior of astrocytes is, at least as far as we understand it for now. And so you can use integrate and fire neurons, and that's fine, because neurons generate spikes, and that's enough. And so it's and you know what you lose in the trade-off. And I think for what we do, it's far less clear for now. And so I think really it should be a main concern until we have a better understanding of neuroglial networks.
And building on that, I want to attack it from a different, another kind of different angle, like building on what you just said. I don't know, I'm not sure if you're familiar with the computational but purely neuronal paper that was published 2 years ago from Idan Segev's lab, actually here in Helsinki in the Hebrew University, which he collected data, experimental data from mice and from human. I mean, he never touched a mouse or a human, but he collected data and tried to compute something out of it. And by the dynamic computation that he did, he found out that it doesn't make sense. And the capacity of humans should be twice as much or half as much. I'm not sure it's something to do with two, but twice as much than, I mean, the difference between mice and humans. And then we went back and found that he found something that no one ever thought about. So from the computational side, we actually understood the experimental view way better.
If I may just add to the Frankenstein part, I'm guessing that we kind of do it because we're taking the parameter values and the data where we can find them. And the thing is that, as Renaud said earlier, we like a lot of data and a lot of databases from astrocytes so that we can have those parameter values that are from astrocytes already. So I think this is one of the main reasons that we are using those data from different species, etc. And I still think we can have relevant information and predictions from those models as soon as they're validated and tested against experimental data, as discussed previously here. I don't think for now we can just say no more Frankenstein models, because we don't really have a choice for now, I think.
Just to add a little bit on the thing, I was finding that I think computational neuroscience as a whole is facing a problem that we don't have a clear consensus on the parameter values for a lot of models. Like, it is a lot of arbitrary, and it seems like one have a broad range of parameter values to choose from, and so that adds a lot of confusion, because depending upon what values you put for one model or one component of the parameter, you might need to change something else. So, and if you have a lot of parameters, then it's like a big problem to find the optimal, and then one has to use some genetic algorithm or complex ways to optimize the model to the data, right? So I think it is very important at some level that people doing modeling at either biophysical or detailed model has to sit together and sort of come with a sort of a rational range of values that's acceptable considering the literature that we have until now, so that when we make new models, it becomes more easy to follow. Because, for example, a lot of models, there are a lot of very good models for astrocytes, but they used a different ER concentration, like they use like 10 micromolars. But we know experimentally that the ER concentration is like 250 micromolars, around that. So if you take that into account, then clearly your parameters for CERCA and the PMCA is going to be way off. So finally the question is, what is the right value? So that seems like a big question, and I think the big problem lies in there, that of course that we all take values from some one literature, and it's very hard to take all the literature into account. But I think at some point we need to sit and sort of find some consensus, and that really helped, I think, to move the field better. And that's what I think. And having more parameters, I think, is a curse and a boon at the same time, because when you want to fit like complex models, it's good to have a real close match with experimental data at some point. But then choosing the right value is the key, right? So, and that becomes a big problem.
other comments? We have one more question, even we are going now a little bit over time already, but I hope you stay with us. So where do you see the challenges of the field, like in short term and long term, and how can we team up together as of course the modelers among themselves, but also especially with experimentalists. So do you have any thoughts on that?
What's this calcium doing? That's that's my 2 cents. And yeah, I actually wanted to start this conversation for the joint discussion between Anup and Rono, but we didn't really have the time. But also the link between this calcium activity and metabolism. I think all of those are very important questions, because for the moment we see those signals and we see correlation with function, but do we really know if they're the cause of the functions that we're seeing, and their role in different physiological conditions? The calcium code, you know, as it's called.
I think I have perhaps more of a comment again about one thing that I appreciated in the models that I have heard discussed in this workshop was the fact that I feel like we are at a point where we are combining the details, say, of subcellular compartments with broad networks, and to me that is particularly interesting, because ultimately we're trying to get to a point where we can understand what a molecule does in the context of a living organism, right? That's the ambition that we have, but that dream can't come true if you don't combine the complexities of single cells within the context of larger networks. So I think one thing that I really was thrilled about here was, you know, knowing about your guys' works about being able to detect the calcium signals in this compartment or in the other, and then integrate them in complex networks. So to me, that was appealing. I think a challenge is the classical challenge, right? How do you as a theorist communicate with an experimentalist and vice versa? There's a little bit of common ground, but I think that's an exercise on both sides. I think there's through initiatives like this that we learn how to do that, not if we keep doing the same thing over and over.
I think we need to find a place to interact more often, because we have the community in CNS that is mostly computational. And I'm super glad that we managed to have some experimentalists with us for this workshop, but it's really unusual. and then we have, so Kerstin and me, for example, at the moment we're at the European GLIA meeting, and there are really, really few computational scientists and very few people that are interested in our posters. And I think it's because we lack those opportunities to make those two fields and those scientists interact and start working together. So I don't have an answer to how we can improve those interactions, but starting with events like this one could be nice.
Perhaps, Audrey, one idea is why not putting together a worldwide neuro channel, something that generates some sort of recurrence. And you can't guarantee that always the same people are going to show up, right? But that's one of the things that I think we have learned from these times, that we have the ability to connect throughout the world, regardless of time zones, and we do our best. But I think keeping that alive, because I mean, perhaps it's a selfish point, but I cannot go to all the conferences that I wish I could go, but I can tune in, right? Like today I'm here in the lab, everything is dirty, but I'm still tuned. I couldn't do that if I had to travel thousands of miles away. So I have personally been active in joining worldwide neuro events, and I wish there was something focused on, I think, like this. So I would be happy if something like that took place.
I'm following up what actually Ala just wrote. We had actually, I hope I can say that, but with Renault, we already had now some discussions to set this up, like to have regularly talks with, like, especially more rather meant for maybe master students and PhD students, but of course we could think how to extend it. And having experimentalists and modelers included in this series. So we have some initial thoughts, but maybe we can actually extend the round. And if you're interested in that, just shoot us an email. So either use like the CNS Gmail address that we have set up, or write us directly. So we would be happy, of course, to hear you there, but also to see you there and to really drive this, because I think also it's very important. I think we're working too much isolated, even like within our field, like within our areas. And I would really like to extend that and use workshops like this. And we were also now thinking what Audrey said earlier, is to maybe go next time to the European CLIA meeting and have it on the other side, basically, where we have more experimentalists and maybe really getting there like a common ground. So I would be like, yeah, I think it would be super exciting. And I think we learn a lot from each other.
Sorry, can you hear me?
Mm-hmm.
Okay, because I couldn't talk before. I don't know what was wrong with my microphone. So I'm going to say something. So yeah, no, I definitely agree. I think it's good to have more meetings. But one thing I've also been thinking about is related to the discussions right now is that so we talk about how there's a lot of parameters in the model. We don't know what those values are and so on. But I think another important point to keep in mind is that experimentally, there's a lot of variability as well, different brain regions, different ages of animals, and different experimental conditions like the sleep, wake, diseases, so on. So I think, I don't know, my perspective personally is that it might be ideal to have a couple of major questions that we want the field to answer first, and to have both experimentalists and modelers focus on those specific questions, because, one person's looking at this brain region, another person's looking at that brain region, all of these differences. And it's hard to have, given that there's not a lot of people working in these fields, it's hard to have enough information for, to have the right model and all of that. So I don't know, that's just my perspective. I think it would be nice to have kind of a couple major questions we want to answer, but then within those questions have experimentalists and modelers kind of lead to that. So maybe in the group that hopefully we can form, the online group that we're discussing, can lead to that ultimately.
I absolutely love the idea, and there might be actually a possibility for that. So I just got the invitation from a journal, so maybe we can even use that. And again, shoot us an email if you're interested in that, and maybe we can write a kind of sort of white paper together and formulate those questions and needs. So yeah, just you can also write on Discord, like just reach out if you want to participate. We're absolutely happy, and as said, like that the connections grow, and also the community. Okay, I think this was a lovely closing word, and for me it was like 2 amazing days. It was super exciting, and I hope that we can, like I said, repeat this, and we will discuss in which way, on what channel, and feel free again to reach out. We're really happy about those connections. We had wonderful speakers, so thank you for taking the invitations, and yeah, we hope to see you soon again.
Are they done?
I thank you all the organizers for organizing this whole talk, and it's a really wonderful organization, and it was a great opportunity for people like me to share the work and learn from a lot new stuff is going on. Good job. Thank you.